All right, let's get started on Chapter 7. We are going to look at Mendelian inheritance, and this chapter is straightforward. You should have had some of this in high school. So this should be review for the most part. But let's go ahead and continue on here. So we obviously receive inheritance from our parents. I mean, it's an obvious conclusion. Exactly how and the diversity of which, that's one of the, or a couple of the areas that we're going to describe now. And so your book talks about your mother and father contribute equally to your genetic makeup. This is almost true, but we'll let it go for now. Of course, there are aspects that being male or female for the offspring, it might throw that genetic makeup equal portion there a little bit off, but that's okay. How can a single bad gene make you smell like a rotten fish? Well, we talked about that in class some, but ideally what we have is certain genes that may have some sort of defect or altered sequence. And some of these enzymes that, in this case, the flavin containing monooxygenase 3, which breaks down a chemical in the body that smells like rotting fish, for some reason that gene is dysfunctional and it can't break it down. So therefore, it's that odor is in the urine, sweat, breath, it excretes it, causing them to, of course, smell like rotting fish. Worst of all, the odor comes from within their bodies. They can't wash it away, and no matter how hard they try, called fish odor syndrome. This odor often causes carriers to suffer ridicule, social isolation, and depression. For those born with this, of course, you can imagine they're going to worry or wonder if they can pass this on to their children. So, of course, this is one of those things that can be passed. And how it's passed, of course, is done through the inheritance of genes. And we have the maternal chromosome pair, which is the diploid, and the paternal chromosome pair, this is diploid, and each of which generate a haploid. You can think of it as half the complement, haploid. Maternal gamete is an egg, for, of course, for the female, and the sperm or the paternal gamete for the male. And during fertilization, gametes unite during that process. Children inherit one set of chromosomes for each of the two parents. So, most of us don't have to worry about fish odor syndrome, but we do worry about many of the other inherited traits. Blame for straight hair, curly hair, the big wavy hair over your eyebrows, stinky feet. Well, I guess maybe, no, that's inherited type of deal from maybe not changing your socks as often as you should or something like that. But anyhow, obviously you can look at traits. Some we see various aspects like the nose or the lips or those sorts of things we do pass along in our offspring. So fish odor syndrome, we can talk about having an egg has the normal version, and then the sperm has the defective version of the FMO3 gene. The gametes unite during fertilization. Of course, the children inherits one copy of the defective version of the FMO3 gene. The child is a silent carrier and will not have the fish odor syndrome. If two copies of the defective version of FO3 were present, the child would, of course, develop it. So this is, it's important to know about your recessive type of traits, and it's just a matter of, for the most part, just asking the various folks that are involved. Maybe their mothers or fathers or something like that. So if you recall from chapter 6, that sexually reproducing organisms generally carry 2 copies of every chromosome. Again, this is the diploid state. At each location on the 2 chromosomes, the pair is the same gene. 1 copy from the mother and 1 from the father. So in this case, the FMO3 would be perhaps a defective version might be on the father's, and in this case, the person would be recessive for that characteristic. But if both parents had perhaps recessive and or were dominant for that particular trait, either having both recessive traits from both parents or dominant trait as a result, might show up as an unlucky catch, I guess, as they say. Then it will be expressed, since there's no normal gene to be expressed. So it would dominate in the offspring. A normal or wild type would dominate from the standpoint that the correct gene would provide an enzyme to break that product down. So that should make sense to you. So offspring resemble their parents. Genes are instruction sets for the biochemical, physical, and behavioral traits, as we know. Some traits are controlled by a single gene. In heredity, passing of characteristics from parent to offspring are done through the genes, as we know. So like father, like son, no one will ever seek a paternity test to prove that Michael Douglas is the son of Kirk Douglas. All it takes is a quick look at their legendary dimpled chins, and it's clear that they're related. We all see that various traits of our offspring resemble some of the traits that their parents do, and passing these on, these types of traits through our families is called heredity. And it's a good idea to ask your grandfather, great-grandfathers while they're still alive, or grandmothers and great-grandmothers. You just ask about traits and various characteristics, because once that information's gone, it may not be recorded, and it's a good idea, unless you want to go into more modern aspects and consider perhaps 23andMe or some of those services to look at your genetics that way. It costs about $100, I believe. So selective breeding, we observe and utilize heredity in our favor. This is not genetically modified organisms. This is just using selective breeding, which farmers have done for a long time. Once breeders recognized the existence of heredity, selective breeding such as increased body size in cattle became possible. And it's just knowing or recognizing that certain plants or animals have certain desirable characteristics, and we cross those to increase the probability of getting those desirable traits. And this is, of course, adds to the value of hogs or the cows or whatever those desirable traits. So in milk cows, of course, having higher output of milk would be highly desirable. So farmers will obviously breed those that have the higher outputs, and then those are highly desired traits, and farmers will pay more for those offspring, of course. So there's an economic motivation for that sort of thing. So are there any human traits determined by a single gene? Traits that are determined by instructions a person carries at one gene are called a single gene trait. Who's buried in Grant's tomb? I mean, that's straightforward. A single gene trait. So mechanisms of heredity are like a puzzle. They become a little bit easier to grasp as we focus on the smallest aspects of family resemblances. Virtually everyone with a cleft chin, for instance, has a similarly dimpled parent. Everyone who has earlobes that are not attached, or the widow's peak, or various things. A lot of these traits are determined by a single gene. So you can look at those for a list as we go on. And if I had hair like the gentleman in the bottom there, like that, I think I would have to pull out a brush or something. But today, a lot of other folks can get away with that. So, how single gene traits like cleft chins are passed on to a child are the easiest patterns of inheritance to decipher. Height, hair color, eye color are a bit more trickier. There's certain things that we need to take in consideration. So more than 9,000 human traits are determined by a single gene. Mendel learned about heredity by conducting experiments. He was a monk that would travel from monastery to monastery, and he had conducted many experiments. So, and he didn't understand genetics. He was trained as a chemist, but really what was important is he was trained with a scientific method. So it wasn't really, in the 1800s, they didn't understand the concept of DNA or any of that. So he was a monk. Now, Czech Republic began some studies that shed light on the questions, but practically answered them. What he did was he melded mathematics with his observation. Now we call that genetics, based on the fact that luckily the traits that he chose with various plants gave him straightforward mathematics, because the genetics were straightforward. From that standpoint, chance favors his prepared mind. Back, Leonardo da Vinci thought that the human sperm had a very, very small little baby inside the head of the sperm, and then it would be planted and then would grow and develop. Obviously, until the 1800s, this was what everyone thought, but this pre-made human idea, it didn't take much imagination to see why that ran into difficulty. Other popular ideas to explain the offspring, various things, they had a really hard time to try to explain it. And Gregor Mendel, of course, made a huge leap in terms of, at that time, our understanding. So, three features of Mendel's methodical research were critical to its success. He used, of course, a very scientific and methodical approach, and using pea plants was a good choice. So the pea plants, they had a lot of things going for them. They're easy to manage. The fact that he visited one monastery to another, he isolated the experiments. All he had to do was pollen dusting, just be able to provide the fertilization this way. He also focused on easy to categorize traits. That would be an excellent idea, and he did that. And then he applied his mathematics and his scientific method to what he did. And that made, plus he wrote it down. Let me make sure that's clear. He recorded what he did, and that's a very important aspect. And that's why through history. So he did a true breeding cross. Now, true breeding is where you cross a dominant and recessive trait. So Mendel crossed a true breeder purple flower with a true breeding white flower. So he knew that every time you cross what he called a true breeding, he always got purple flowers, and same with the white. And when he crossed those two, he came up with some different types of variants, and this was important. So true breeding purple flowers always produce purple flowers offspring, and the white with white. But when he did the cross, he got some surprises, and we're going to talk about that. So let's recap. Gregor Mendel conducted studies in the 1800s. He used the scientific method, and he focused on easily observed and categorized traits in garden peas. So, question. Mendel figured out the rules of genetics by working with pea plants. Why were these plants a good tool for Mendel to use these questions about inheritance? Individual plants could be self-fertilized or cross-fertilized. They grew quickly, short generation time, and clearly defined traits. And boy, those read like all good things. And if you said 4, you would be right. So 4 would be the right answer. Pretty straightforward. Segregation, you've got 2 copies of each gene, but only 1 copy in each sperm or egg. The odd and recurring results spurred Mendel to figure out the mechanisms by which traits could be passed from parent to offspring. As brown-eyed parents would have blue-eyed children, sometimes traits weren't present in either parent or the pea plant that showed up in their offspring. So crosses that entailed the fertilization of plants with purple flowers by pollen from the other plants with purple flowers produced mostly purple, for instance, but sometimes they produced plants with white flowers. How was this possible? So going back to the experiment where he took the true breeders and crossed them, he found that all the purple colored flowers were in the offspring. However, if he crossed those flowers in the second generation to create the third generation, he found a mixture of the purples and whites. So this concept, a recessive trait, which he did not coin at the time, he didn't quite understand what was going on, but a good observer will write down exactly what he sees, and he applied his math and his methodical reasoning to it. And that was clearly the gift that Mendel brought to this process. Here is where Mendel's meticulous methodical experiments paid off. He started with true breeders. He got some true breeding purple flowered plants. He wondered which color wins out. So when he crossed them, all the offsprings were purple every time. For this reason, Mendel called the purple flower a trait dominant, when he considered the white flower trait to be recessive. So he understood through observation and came up with a theory or hypothesis, and in general, a dominant trait masks the effects of a recessive trait, and he could support that with his math. And this makes for very good science. So according to Mendel's law of segregation, this is a really important one, only one of the 2 alleles for a gene is put into a gamete. At fertilization, offspring receive from each parent 1 allele for each gene. So heterozygous pea plant with 2 different alleles, white, purple, for the same gene flower color, and heterozygous pea plant. So you have 2 heterozygous, so that means that hetero means different. So through meiosis, each gamete gets 1 copy. So each now of those parents would produce either a white allele or a purple allele. Now, allele means that it's a trait for the same gene. So a white flower or a purple flower, so that is an allele or that characteristic. So we'll come back to that if you didn't quite follow me on that. Fertilization, each fertilized egg gets 2 copies, of course, and 1 from each parent. So at some probability they get either both of the recessive or the white, 1 white, 1 purple, 1 purple, 1 white, or 2 purples. And if you look at how this is crossed, you can just look at the frequency, and this is played right to the strengths that Mendel had. So when he took the purple flowered plants and came from the cross of a purple white flowered plants, he found that the mixed parentage plants were no longer true breeding. They would produce the white flowered offspring. So this is what he came up with as far as his hypothesis to explain what he saw, is his laws of segregation. And this is that was pretty cool what he did. And I must say, for a monk, I guess he didn't have TV or Xbox games to distract him, but anyhow, 3 ideas that Mendel used for explaining this pattern of inheritance. Each parent puts into every sperm or egg, it makes a single set of instructions for a building of a trait. And this helps explain what's going on in terms of, rather than passing on a trait itself, each parent puts into every sperm or egg, it makes a single set of that instruction for that building of that trait. So that is the instruction set we call a gene. He didn't call it that in those days, but that's what it was. 3 ideas. The second one is the offspring thus filled themselves with 2 copies of instructions for any trait, which we call allele. So there's a good definition. An allele is a copy of instruction for a particular trait. So an allele might be various variations of that trait, but it is a derivation of that trait. It's not too arbitrary. So offspring thus find themselves with two copies of instructions for any trait. Most of the time it's the same, but sometimes it's different. Often both sets are identical, but the alternative forms are called alleles, and there we get the variations that we know about. So the third one, the actual trait produced by an individual, depends on the two copies of that gene which they inherit from their parents, homozygous and heterozygous. So the actual trait produced by an individual depends on the two copies of that gene they inherit from their parents. So homozygous shows the trait by the instructions embody those alleles. There are two copies of the same, the homozygous, homo being the same, and hetero being different. So we have a dominant recessive alleles determined by these different alleles, and we call that heterozygous state when we have that alleles are fully apparent, the individual with the other is not. So the first one's called dominant, which masks the effects of the second allele, or the second part we call the recessive allele. So you can see this clearly if we go back to that figure 7-9 and look at the assortments there and the possibilities. The lines that they have drawn there are a little bit confusing, but if you follow them, that will come apparent. So we actually have ways using Punnett squares to help delineate this, and we're going to work with some of those just to try to help organize this instead of drawing lines like that. It gets a little confusing, and that's the key to working out these types of problems. So when offspring grows up and gets ready to have its own offspring, it only gets to contribute one of the two copies that it received, or the baby received originally from its parents. So the other parent will contribute the other gene. So when the sperm and eggs are made, each of these sex cells only get one copy of the gene. So as opposed to the other two copies of every cell in the body, for a male who is heterozygous, it means that half of the sperm he produces will have one of the alleles and the other half will have the other. The idea is that the two copies of the gene everyone carries, only one of those two alleles gets to go as a gamete. So this is Mendel's law of segregation. And he just observed this mathematically, and that's where he came up. Obviously, they're getting segregated. So each parent puts a single set of instructions for a particular trait into every sperm or egg. The instruction set is called a gene. The trait observed in the individual depends on the two copies or alleles of the gene it inherits from its parents. Observing an individual's phenotype is not sufficient for determining its genotype. So a phenotype is what we observe, little or no pigments in the eye, hair, or skin. The genotype, it's either homozygous or recessive for an allele, like being an albino or albinism, and you can see that white one sticking out there. So things are not always as they appear. The skin coloration, for example, humans and many other mammals have a gene that contains information producing melanin, which is a pigment, one of the chemicals responsible for giving our skin coloring. Unfortunately, there's also a defect, non-functioning version of this melanin gene, which is passed along in some families. An individual inherits two copies of this defective version of the gene, cannot produce the pigment, and this condition is called albinism, a disorder characterized by little or no pigment in the eyes, hair, or skin. It's impossible to tell which normally pigmented individual carries one of the defective alleles just by looking at them, but through genetic analysis one might be able to determine this. So the outward appearance of an individual is called the phenotype. It's something that we see. Underlying the phenotype is the genotype. So the gene, hence the name genotype. Pheno is a Latin term becomes apparent, or something that you become to know, or we know as a group. I know it's kind of vague, but that's where pheno comes from. So that's an organism's genetic composition is the genotype. We can try to predict from the genotype what the phenotype might be, but we're not always right about that. The inability to deduce an individual's genetic makeup, though, is a simple observation, is generally a problem in genetics. Physical appearances don't always exactly reflect the underlying genes. A normally pigmented individual may have 2 copies of the pigment-producing allele, or they may only have 1. In either case, individual will look the same. The outward appearance of an individual is called the phenotype. A phenotype includes features visible to the naked eye, such as a flashy coloration, height, or the presence of antlers. A phenotype also includes easy visible characteristics such as the chemicals an individual produces clot blood or digests lactose, lactose intolerant. Individual's phenotype even includes behaviors. Underlying the phenotype, of course, is the genotype. This is the genetic composition. This is what, of course, Gregor Mendel had no clue about, but he was using the phenotype to be able to make some generalizations about how things segregate. For example, an individual's genotype is described as homozygous for the recessive alleles for albinism. So it has 2 copies of the recessive allele, or the gene for no pigment or albinism. Another individual's genotype for melanin gene might be described as heterozygous, having 1 copy of the melanin gene and the other copy not producing melanin, and the dominant trait producing melanin will win out, and that is heterozygous, and it would become a dominant trait. Occasionally, the word genotype is also used as a way of referring to all of the genes of the individual carries. So you have to look at what context the word is used. It's pretty straightforward, not to worry about. So genotypes, we have homozygous, dominant, and heterozygous. So when an organism exhibit a recessive trait such as albinism, we know that certainly the genotype is. We show is that when it shows the dominant trait, on the other hand, it's impossible to distinguish an organism who carries a dominant allele, homozygous dominant, or carries 1 copy of the dominant allele over the recessive heterozygous, because it's not possible to visually discern the genotypes of the 2 individuals with the same phenotype. Much of the genetic analysis there must make use of clever experiments and careful record keeping in order to determine the organism's genotype. Well, I think this dates our textbook a little bit, because now we're getting into techniques by simply scraping a buccal cavity or your mouth and send that into companies. It's going to become more and more likely that they can discern those things using genetic techniques. So how do we analyze and predict the outcome of crosses? Assign symbols to represent the different variants of a gene. Generally, we use an uppercase letter for the dominant allele and a lowercase for the recessive allele. So that makes good sense as far as our doing good paperwork. So how do we analyze and predict the outcome of crosses? First, we must assign symbols to represent the different variants of a gene. Generally, we use an uppercase for the dominant allele and a lowercase for the recessive. We represent the albino individual as AA because it must carry 2 copies of the recessive allele, A. An individual trait that's pigmented must be either AA or A little a. If we don't know which are the 2 possible genotypes, we usually just put a blank after the major big A, where the placeholder is the unknown second allele. We just don't know yet, and we have to run crosses. So here is a Punnett square looking at the mother as an albino. So we know that the gametes that she can possibly deliver is an A, little a, and a little a. And the father is pigmented, homozygous. And we know then the father is big A, big A. So all the offspring of this cross would be big A, little a. And that is 4 to 1. If the mother is pigmented and the father is pigmented, heterozygous, and the mother is heterozygous, and then we look at the fallout of the particular gametes, then we see big A, big A, big A, little a, big A, little a, and little a, little a. So the phenotype, both would be 3 out of 4 would be pigmented, and 1 would not be. So this is the math that Gregor Mendel would follow, but genetically, we look at this as we see 1 to 2 to 1. In the phenotype, we see 3/4 or 75% are pigmented to 25%, or 3 to 1 for the phenotype. So 1 to 2 to 1 versus 3 to 1 on the phenotype. If you follow me on that, then we're almost there. So we can trace all the possible outcomes using a Punnett square. We illustrate the cross between true breeding pigmented individual PP with a albino p. So on top of the square, we list individually the 2 alleles that the one of the parents can produce, and on the left side of the square, the 2 alleles that the other parent can produce. And we have to know, are they homozygous or heterozygous, and that sort of thing. So we split the individual 2 alleles, remember the segregation, up because although the individual carries 2 alleles, there's only one of the alleles for each sperm or egg. Remember, haploid or half. In the four sections of the Punnett square, then we enter the genotypes of the possible offspring, is illustrated what we just saw, and every possible offspring would be heterozygous or would be normally pigmented because it receives a dominant allele from the pigmented parent and recessive allele from the albino parent. So we can trace the cross between the heterozygous individuals. Note that each parent produces 2 kinds of gametes, one with a dominant and one with a recessive. This cross would have 4 outcomes. 1/4 of the time the offspring would be homozygous. 1/4 of the time the offspring would be homozygous recessive. Homozygous dominant and then homozygous recessive. I hope I said that right. And the remaining half of the time the offspring would be heterozygous, big P, little p, or phenotypically 3/4 the offspring would be normally pigmented, or 1/4 would be albino with a homozygous recessive, or little p. And if you follow me on that, then I feel like you've gotten what you needed on this particular section. So it's not always possible to determine an individual's genotype from its phenotype. A recessive allele's effect may be masked by a dominant allele. Genetic analysis makes use of clever experiments in Punnett squares. So I'm going to give you a few Punnett squares to work in class so that we can get a good understanding. So we'll stop here and let you think about how these worked out, and give you a chance to work through some problems, and then we'll pick up next time on probability and chance playing central roles in genetics. So I appreciate your time and your diligence, and we'll continue on now with the next section as soon as we can. So I'll see you in a little bit.